Uh, First of all, thanks to the organizers for inviting me back to give a talk at HCA Asia. I gave a talk in Singapore last year at this meeting, and I briefly mentioned this work that we were doing at the time called scONE-seq, which is a technology for simultaneous profiling of DNA and RNA from single cells so that we can obtain both the whole genome and also the transcriptome from the same single cell. And I'm very happy to present a little bit more on this work this year. With this method, hopefully we can share with everyone soon so that you can also use it. So I guess with this group, I don't think I need to say too much about why single cell multi-omics is important. Even not at the single cell level, people have already demonstrated that when we can access multi-omic levels of information, we can address unanswered biological questions. And I think for studies of cancer in particular, this has been demonstrated by the pan-cancer multi-omic profiling effort that was published in 2018, which is what I'm showing here. In particular, in this pan-cancer study, you can see that if you only look at the mRNA, actually, it doesn't give a very good subclustering of different cancer types. It's only when you layer the mRNA, the DNA methylation, and the copy number information that you can really get informative subtypes of cancer that inform the therapy as well. So this is sort of the motivation for why multi-omics is becoming more and more of a pressing issue to be solved on the technology side. And of course, applying it to single cell will be particularly useful for uncovering heterogeneity. So I'm certainly not the first person to try and develop a single cell DNA and RNA simultaneous profiling method. There have been other methods developed, and generally I categorize them into two types of strategies. So the more popular one is this separate strategy, which I'm showing here at the top. The separate strategy essentially requires some method of physically separating the DNA and the RNA. So on the left here, GNT-seq, this particular method was invented around, I think, 2015. And what they do is use a poly-T functionalized beads to pull out the poly-A RNA from a single cell lysate and then reverse transcribe and amplify those separately from the genomic DNA. Professor Tangfu Chou already showed his work using their method, single-cell trio-seq, where they physically separate the DNA, or they physically separate the nucleus and the rest of the cell by centrifugation, and then do both methylation studies and RNA on the same single cell that way. He has obviously shown that you can get a lot of very interesting data as well from this method. And a third method that was published more recently in 2018 called scNC-seq uses dielectrophoresis in a microfluidic platform to, again, lyse the outer membrane and leave the nucleus such that the cytoplasmic RNA and the nucleus are separated, and the DNA and the RNA can be analyzed separately in this system. So these are all methods that use the separate strategy, where there's some selection of DNA and RNA and separate the two and process them separately. There is one other method published called DR-seq, which uses a split strategy. So they do some preliminary amplification of the RNA and then just split the entire pool into two halves and then do RNA processing on one side and DNA processing on the other side. All of these methods are, of course, viable, and they're feasible and they work. There are, however, a few remaining sort of limitations to these existing methods. As you can see with the separate strategy, you need to be using either beads or centrifugation or specialized microfluidics, which can pose adoption difficulties, or in the case of sort of bead-based or like pipetting-based methods, they can be difficult to automate and scale in terms of the number of cells that you can analyze. The split strategy could be easily scaled up in a well plate, but because you're splitting it, it does suffer from more dropout issues. And overall, all of these are difficult to implement on frozen samples, because with a lot of frozen tissues, you can only obtain the nuclei. For example, in the brain, you can only obtain the nuclei, and it's very difficult to perform any of these on that. So we really wanted to come up with an approach that would kind of address some of these remaining challenges and be a very generalized and scalable method. And so our method that we've developed for simultaneous whole genome and transcriptomics is called single-cell one-seq. And the sort of I think the most attractive part about one-seq is that it is a one-pot reaction, so there's no need to centrifuge. We don't use any beads. There's no sample splitting, no microfluidics, so which means everything is made to fit a standard workflow in the lab, especially for single cell. It's made to fit a standard flow cytometer-based workflow, and because it's plate-based, you can automate it to be done with a liquid handler. And so the rough idea is that we still put single cells into individual wells, and I think it could be compatible with the seqWell platform as well. You put cells into single wells and then lyse them, and then we do DNA fragmentation and barcoding to add a DNA-based barcode. And then on the RNA side, we do the RT and add an RNA barcode. And these all happen in the same reaction. And then once the barcodes are attached, we amplify the DNA and the RNA simultaneously. And then we just construct the library from that as usual. And so this is, again, like the specific workflow. As you can see here, everything happens in sequence. So you just continue adding reagents to a one-pot reaction, and there's no physical separation of the DNA and the RNA. And this protocol was developed by my student, Yulei, who's shown in the picture here. Also, you might notice that actually in this reaction, we are using a random priming plus a poly-A plus random priming based RT approach. So that means that we are amplifying the total RNA and not just the mRNA, because we really wanted this to work not just for mRNA. We wanted to be able to look at frozen tissue and be able to analyze nuclear RNA as well. And so that means that we would be, in the usual case, capturing also a lot of ribosomal RNA fragments. And so to address that, my other student, Danson, developed or adapted the original DASH protocol using the CRISPR-Cas system to be able to deplete ribosomal RNA sequences from a single cell prep. And so using this modified DASH protocol, we're actually able to deplete most of the ribosomal and mitochondrial RNA from our single cell libraries. And you can see from the left here, the results. After our DASH treatment, the amount of ribosomal RNA in the library is greatly reduced, and we can profile a lot more mRNA in the sequencing output. So actually, this is a manuscript that's currently under review, and again, we hope to make this protocol available for everyone to use very soon. Okay, so what are the results from the single cell one-seq look like? We first did some validation studies just looking at HCT116 cells. So we captured transcriptomes from 96 single cells using our one-seq method, and then also 96 cells using SMART-seq2, and we did a comparison in parallel between the two methods. The takeaway here is that after using our single cell DASH protocol, the amount of transcripts or features that map to exonic, intronic, and intergenic regions is roughly comparable between the single cell one-seq and the SMART-seq. The only thing is that we do have more sort of unprocessed RNA fragments that we capture since it is a total RNA capture method. And in general, the total number of genes is also higher for the one-seq method because of the capture of non-poly-A RNA. Generally speaking, we also have a comparable dropout probability compared to SMART-seq2. So the sensitivity of the method is very comparable to the commercial gold standard. And as expected, because we're using a non-poly-A method of capture, we also have more coverage at the 5' end of the gene bodies. So on the RNA side, it looks like our method is pretty comparable to the gold standard. We also then looked at 4 different cell lines. We profiled 4 different cell lines using the one-seq method, and we wanted to make sure that we could capture DNA copy number changes at the same time as capturing mRNA. So here I'm showing you all of this data is paired DNA and RNA, and on the left, on the RNA data side, you can see that the 4 different cell lines separate out very cleanly, whether by visualization using a UMAP method or in terms of the heat map, differentially expressed genes from the different cell lines, and then major marker genes are also captured for each of the cell lines. And on the right here, I'm showing you the whole genome sequencing data from these 4 cell lines. Oops. from these 4 cell lines. And the takeaway here is that you can see from the single cell data, the top 2 cell lines, MPC43 and HCT116, are both cancers. And you can clearly see the sort of genomic amplification and deletions in terms of the copy number changes in those cell lines, whereas the 2 other cell lines that we profiled, the H9 and the HUVEC, are both normal cell lines, the H9 being an embryonic cell line, and the HUVECs are an endothelial cell, vascular endothelial cell line. And both of those don't show any significant deletions or amplifications, indicating that the whole genome sequencing data is also quite reliable. We've yet to validate this, we've yet to show a clear validation with the bulk, which is currently what we're doing to further verify that the DNA side is indeed reliable. But overall, compared to mini bulks, so far it looks like our data is comparable to a bulk sequencing when the single cells are all merged together. So after doing validation on cell lines, we wanted to make sure that it works on primary cells as well. And the first thing that we did was sequence a bunch of PBMCs. Here we did 190 PBMCs from whole human blood. And from this, on the transcriptome side, once again, we can separate out the different cell types. And using the different markers that were detected, for example, we can distinguish the T cells from the B cells using the canonical CD3 and CD79 markers. Using CD4 and CD8 expression, we separated out 2 different classes of T cells that we captured in the PBMCs. Then we can further identify effector CD8 T cells specifically in this sort of small tail down here, using the, again, known markers that are distinctly expressed in that subtype. And finally, we also identified a group of NK cells in the PBMCs as well in the tail here. So it looks like the transcriptome data from PBMCs looks quite good on primary patient or primary human samples as well. Of course, we compared that to SMART-seq2. So this is the data from SMART-seq2. And actually with the SMART-seq2, it somehow did not end up capturing any CD8 effector T cells. It's hard to say whether it's because of the data quality or because we captured total RNA and we're able to see a distinct signature compared to SMART-seq2. But overall, the agreement is quite good. Because this is from a healthy donor, so there was actually no copy number changes observed in the DNA side of this data. Okay, so let's get to the interesting stuff that we can do with this method. One thing is that we can use ONE-seq to study host viral interactions. So we applied this method to nasopharyngeal carcinoma cells, MPC43. This is a cell line that was established from a primary patient tumor. And we were interested to see what is the heterogeneity of this cell type, because actually the EBV that drives the nasopharyngeal carcinoma is actually heterogeneously expressed inside these cells. And so it's after using the single cell ONE-seq to profile MPC43 cells, we noticed that we could capture both the endogenous human transcripts as well as the viral transcripts, and also both the human genome and the viral genome as well. And the paired DNA and RNA gives us the ability to separate out important cell types. So EBV establishes latency inside the MPC cells, and that means that there are some cells in the MPC which are EBV negative, meaning there is no viral genome at all. But there are also EBV positive cells which are either latent, meaning that they express very low levels or no EBV RNA. And then there are the cells that are in the lytic stage, which actively express the EBV transcripts. And so you can see from these 2 graphs on the left here, first of all, we notice that the number of reads we detect from the EBV is not correlative with the sequencing depth, meaning that it's not the quantity of EBV reads that we detect is not because we sequenced more, and therefore it generally shows that it's not limited by the sensitivity of the method. And then if we correlate the EBV genome reads to the EBV gene expression, you can see that in general, the correlation is positive. So the more copies of EBV we have in each cell, the higher the expression level of EBV genes also in that cell. But as I show here with this red circle, highlighted in the red circle, these cells clearly have a high quantity of EBV genome copies, but a very low or lower than expected EBV gene expression. And so we would probably say that these are likely to be the EBV positive latent cells. And whereas on the corner here, both genome copies and EBV RNA copies are low, these are probably cells that are just EBV negative. They don't even carry any of the EBV genome at all. On the right, we're showing the RNA results from that. So from the RNA results alone, you can't really tell which ones are EBV positive and which ones are negative, as the EBV expression only is, the gene expression is only active in a subset of cells, but really there's the EBV genome is existing in a larger number of these cells. And again, we can also look at the genome of the MPC. So this is the copy number variation that we can see for the human genome in the EBV cells. And you can see that compared to at the top, in the black and white, this is the published the published MPC43 chromosome information. And you can see that the copy number changes that we detect in the single cell data matches quite well to the copy number modifications that are detected in the bulk. And of course, the nice thing with the single cell data is that you can detect subclones. As you can see visually, there are definitely some groups of single cells that have acquired additional mutations or copy number changes. So that's just an example of what we can do in terms of viral and host interactions. As I mentioned, single-cell one-seq also differs from other methods in that we can apply it to frozen tissue by doing single nuclei sequencing. And so what we've done here is taken frozen brain tissue. This is a glioblastoma sample. We made single nuclei suspensions from this frozen tissue, sorted them into plates, and performed single-cell one-seq. And again, we are able to obtain both the transcriptome data, which gives us the cell type classifications, as well as copy number changes, which gives us the clonal information as well. So this is just to indicate that this method is indeed applicable for frozen tissue. And this, again, is very useful as a lot of the clinical samples have been banked as frozen tissue. And so this particular method, the one-seq method, will be able to make analysis of those frozen tissues possible. Oops. Before I end, I just wanted to mention one other project that we completed. This is an open source software for integrating single-cell RNA-seq data from multiple experiments and platforms. It's called, the software is called FIRM, and this was developed in collaboration with Dr. Mingjing Si and Professor Yang Can from HKUST. The software is available on GitHub, and you can find information on this software on our preprint publication. So if you just search for FIRM, fast integration of single-cell RNA-seq data, you should find this bioRxiv paper where all the information is. And in there, we show that our method is in some ways superior to the standard sample or data integration method that most people are using on Seurat. I really hope that more people will adopt this method or at least try it out and let us know whether you like it or not. So that's the end of my talk. I think I only have 15 minutes, so I try to end on time. This is my group. You've already seen Yulei, the student who basically developed, single-handedly developed the one-seq method. And I just wanted to also thank our collaborators, Professor Wang Jiguang, his student Wu Xuanhua, and Dr. Danny Chen, who provided the glioblastoma sample. And also special thanks to Professor Huang Yanyi and Wang Jianbing in Beijing. Of course, also my funding sources as well. And thank you. I'm happy to take any questions.
Okay, excellent. Excellent, Angela. Very interesting talk. So I have a couple of questions from the audience here.
Sure.
One is, how do you match the DNA and RNA profile of the single cell? Are they using the same barcode?
So the cells are still individually sorted into well plates, or I guess you could do it in a seq-well kind of format as well. The barcodes are the same across the different plates, so you are processing the plate wells in parallel, still separated from each other. So there's no pooling until the very, very end when you have a a cell barcode as well. So yes, but we are actually developing a next generation version of the ONE-seq right now so that we can pool the cells at an earlier stage and then further increase the throughput. But that one is certainly not ready yet to be shared. Yeah.
Okay, so we have another question here from Ram in Singapore. This is a beautiful talk, Angela, and very impressive technology. And he is asking how different is a host transcriptome in the EBV latent versus lytic cells?
Yeah, thanks for that. That's a great question. So currently, as you can see, we've only collected, I think for the MPC, we only have around 300 cells, which is not quite enough to capture enough to compare the latent versus lytic, but preliminarily, so we have, I think, captured in terms of the transcriptome signature, the latent cells, we captured maybe 20 or so cells, and indeed they express more of the latency associated marks or genes expressed by the EBV. In addition to that, we also see some cells that have very low EBV gene expression, but express a lot of the early lytic genes rather than, and none of the late lytic genes, which suggests that among all the EBV positive cells, there is this sort of gradual increase of transition, from latency to lytic phase. And so currently we're doing more on this project. For example, we treat the cells with drugs to induce lytic activation and then see if we can see that transition stage between the latency and the lytic stage.
Okay, so I'm going to combine two questions here, one from Jasper Huang and another one from Andrew Farmer. And then these questions are, how many genes do you detect per cell? And then can you apply these to methylation?
Okay, let me answer that. So how many genes are detected? So currently with the ONE-seq protocol, we actually find that depending on the quantity of DNA versus RNA barcode that we add, we can actually bias the results toward either getting more higher quality RNA data or higher quality DNA data. So let's say if you are really interested in this new cell type and you want more RNA information and you just want like a rougher version of the DNA copy numbers, you can bias the bias the RNA quality, and we can get around 13,000 to 15,000 genes, assuming that you sequence deeply enough. But in the glioblastoma example, because we're only sequencing the nuclei, there's a lot less RNA in there. So that one, I think we did a very shallow RNA sequencing, only about 50,000 reads. And I think the number of genes we detect there is much lower, like maybe 5,000 or so, but it's enough to give us the cell type classification already. And in that case, because it's a tumor, we cared more about the DNA copy number. So we got a much higher quality DNA data for that data set. The second question was whether we can adapt it to methylation. We haven't tried. I would need to think about how the workflow would work because we really wanted to aim for this one-pot reaction. And I think to do methylation studies, usually it cannot be in a one-pot reaction. So I will have to think about it a bit more, but currently the answer is no.
Okay, one more question. How about the DNA copy number resolution?
So currently all the data that I've shown, we've used the 500 KB bin. We've yet to, like, we need to do the sort of trying on different bin sizes to see what resolution or, what resolution it gives us in terms of the copy number variations. I will admit that because I have more experience in RNA, so most of the characterization we've done so far is more on the RNA side, and we're still trying to pick up the validation and give more detailed specs on the DNA side. But so far it looks like 500 KB bins work well. We may be able to go a little bit lower than that, but probably not much lower. And it's quite comparable to like GenomePlex or WGA type of methods.